Can everyone see this again, or am I not sharing the right screen?
I can't see anything.
There we go. Is that good?
Yeah.
All right, sweet. So we talked about these different species, hybrid zones, how they're coming together. Now we're going to talk about how scientists look at the evolutionary history of a species or a group of species. What are the techniques they use? And how do you look at phylogenetic trees and things like that? They can be very overwhelming if you don't know much when you're looking at a paper. And even if you know a lot, they can still be very overwhelming. So I wanted to go through this a little bit more slowly, just in case. And so phylogenetics, again, is the evolutionary history of a species or a group of species and how we compare them. So, as we go through science, we're constantly learning stuff and we're constantly updating this tree of life, right? So the tree of life continues to change. Right now we have 3 domains of different types of cells that we talk about in terms of being the primary forms of life. That is something that has changed even in the last 30 years, right? We've added an entire domain. If you were in school a really, really long time ago, you would have learned that we split the world into pretty much just plants and animals and then bacteria. But we've actually come to realize that it's a lot more complex than that. And so now we have what are known as 3 domains, and it's based again on the type of cell the organism has. So the first one that you probably know of is eukarya, which is us, right? It's plants and animals, all of those cells. And so we have plants and animals. Then we have bacteria, which you've definitely heard of. Staph infections, all of that good stuff, gram-positive. We looked at bacteria in the lab in 108, right? We plated them. But then there's this cool idea of archaea. So I don't know if you know about archaea, but they are small single-celled organisms that are very similar in structure to bacteria, at least compared to eukaryotes versus bacteria. And I'll talk about them on the next slide of why they're cool. But we have it split into 2 kingdoms and 3 domains. And so what we've been doing now is, as I said before, right, we used to do all of this analysis based on physical traits. Now we have really cool genetic information that we can look at, right? We did that in lab. We took bacteria. We took out their what is called 16S ribosomal DNA. So it's pretty cool that we share a lot of the same base materials, enzymes and proteins with a lot of organisms, right? Most living things have this same structure of their ribosomal DNA, right? Everything needs to make proteins. And so one thing that we do nowadays is we take that primarily conserved sequence, the 16S ribosomal DNA, which if you're thinking about ribosomes, the 16S is that smaller portion of the ribosome. You remember that there's 2 that come together in order to create your proteins. And so when in lab, we took out DNA, we used PCR in order to amplify the 16S region. And so we compared those between species, and right, we use that database in order to look at what species have slight changes in that ribosomal DNA. And so what we found out is that, it seems that the group between bacteria and archaea, we all split apart about 4 billion, between 2 and 4 billion years apart. It's a very large range, but it was also a long time ago. And so archaea are really cool because they're known as extremophiles. And an important thing about archaea is that you actually used part of archaea in lab. So remember when we were running PCR, the way PCR works is we have these cycles of heating up the DNA, and the DNA splits apart. And then we cool it, and then we have DNA replication occurring, right? Well, if we were to use our own enzymes from DNA replication in order to use for PCR, they would denature, right? Our enzymes are made to work in our body temperature, not in the extreme heat of PCR. And so if we were to use our own Taq polymerase and things like, or our own DNA polymerase, it would denature and not work. So what do? We actually found a species of archaea that lives in thermal vents and lives in extremely hot environments. And so they're used to doing DNA replication in those extreme environments. So what do? We use archaea DNA polymerase, and we put that into the PCR machine. It can stay alive and stay healthy and properly function during those heat cycles. And so we actually are using archaea to conduct science already. So they're extremophiles. They're known for being able to handle the most extreme environments on Earth, extreme cold, extreme hot. In this case, this beautiful, colorful lake here is actually an acid lake. And so archaea can live in there and actually live off the acid and survive in these environments. And so they're really, really cool for that reason. And so they're also single-celled organisms. And one thing that's also strange about them is they're non-pathogenic. That we know of. We haven't found a single species of archaea that seem to make humans host and seem to make us sick. Perhaps there's some we haven't noticed, or perhaps they work with bacteria, but for now, archaea are pretty harmless to us, which are really, really nice. But they also come in these unique shapes. They're super small. They're between 1 and 10 microns. They're even smaller than bacteria, or the size scale of the smallest bacteria. But they come in these strange shapes like that. They come in perfect circles and triangles and like these very, very unique patterns. And one of the ways they're able to do that is they actually have a much sturdier cell wall and cell membrane than eukaryotes and bacteria. When you look over on this picture on the right, I'm not going to ask you, I'm going to ask you probably something about, isoprene versus non, but if you look at these cell walls here, eukaryotes and bacteria have these phospholipid bilayers, right? We already know that. We have these lipids, these fats that come together, they form this bilayer, and they protect us, right? In archaea, their phospholipid bilayer is actually a lot more rigid. And so instead of having this bilayer, they actually have a connection between these layers, which may seem like a small difference, but it actually stabilizes a lot. Also, in their linkage up here, eukaryotes have what are known as ester linkages with these double bonds and all this oxygen connected to glycerol and phosphate. Archaea instead have isoprene and ether. And so I'm not going to ask you to know differences and like identify these structures from a graph or anything. I just want you to know that archaea have a much more stable cell wall and cell membrane due to the incorporation of isoprene and also the change of these different links. And so it allows them to live in environments that we absolutely cannot. Sorry if I go a little quick in the beginning. I'm totally forgot. People are like writing notes now. I have to get used to that again, as opposed to people being on the computer. So this video just talks a little bit about extremophiles in general, because one thing that's cool is though archaea are known as the domain of our extremophiles, like all of them do that, extremophiles actually do exist in every domain of life. There are eukaryotes who are extremophiles and plants and et cetera and so forth.
Intense heat, freezing cold, high acidity, and radioactivity. These harsh environments don't seem hospitable for life, but some organisms not only survive, but thrive under such extreme conditions. The name extremophile means extreme lover. These organisms live in exceptionally harsh environments, such as hot hydrothermal vents or buried in rocks far beneath the Earth's surface. Extremophiles occur in all three domains of life: bacteria, archaea, and eukaryote. They range from the extreme heat-loving thermophiles, which feed off of inorganic chemicals and have special enzymes to survive high temperatures, to extreme cold-loving psychrophiles, which have evolved antifreeze proteins that help ensure their survival in some of the coldest waters on the planet. But the most extreme living things on Earth are tardigrades. Also known as water bears, these water-dwelling microanimals are polyextremophiles. This means they are capable of surviving multiple harsh conditions. They are nearly indestructible and have even survived the extreme conditions of outer space. Tardigrades have a unique adaptation that allows them to curl up into a dry, seemingly lifeless ball and slow down their metabolic rate. In this state, they can survive cold, dry environments, like space, for decades. Studying the adaptations of extremophiles may hold the key to solving many of Earth's problems. This includes the development of genetically based medications, producing new types of biofuels, and protecting people against radiation exposure. Extremophiles have opened our minds to the many possibilities of life and the environments that can support it. Understanding the limits of life in these extreme conditions on Earth may provide scientists with clues of how life could possibly exist elsewhere in the universe.
Right, so we have these archaea that are being extremophiles. We're utilizing them for our own science, which is really nice. But how are all these organisms related, and how do we know about that? Right. And so you already know this to a small extent, right? At least, we break down organisms into different classes, right? We have species, genus, family, order, class, phylum, and then, kingdom and domain, which we already discussed, right? And so when we talk about each of these levels, we're talking about getting more and more specific in terms of shared traits between those organisms, right? When we first did this, it was all, morphological comparisons. But what we've learned is, there's a lot of what's known as, convergent evolution, right? The same problems can be solved in similar ways if they're even if they're in completely different environments. So you have organisms that look very, very similar, but can be very distantly related, right? There are many species besides just, say, cactuses that are plants that have learned to live in dry environments. And so domain is the most vague and has the most broad definition for what these organisms are. Species is most specific. And so generally when we talk about these, we try to use the binomial method. So we do species and genus. And so notes about that is species and genus are always italicized, which is how you can tell what they are. If you see these other things like family, order, not italicized. Generally speaking, when you talk about an organism, you only need to tell the binomial name of it, right? So what is the capital letter? Capital letter for species, and then genus. And then we can kind of immediately jump down to this level instead of, slowly inching our way forward. And so again, as we move down, any of these groups, we're getting more specific and have more similar traits. And so any specific group at any level is a taxon. So you can, and so this is one thing about phylogenetics that gets a little confusing is terminology that will be the same. So talking about the taxon of the class of Mammalia, so mammals, you'll kind of have the same context in talking about the taxon of the genus of Panthera. So you need to make sure that you understand when you're reading things or looking what specific taxon they're looking at. And so does anyone see any innate issues with like this setup that we're using in order to describe organisms? Any complications that might arise, perhaps? So what I was thinking of is the fact that, you know, as more species come to exist, as we discover more species, we're going to find areas in which, you know, species don't exactly fall into these perfect clear-cut subgroups, right? Life is a lot more complicated than we could ever just talk about these, you know, these 12 groups, right? So for instance, the organisms that I study a subfamily of spiders. And so when we get down to these more specific groups, you start having things like subfamilies, subgenuses, start having things like tribes and other ways of describing physical traits. And so, you know, we came up with this system a long time ago, but it can get much more complicated than this. And just because two things are in the same family or the same order does not necessarily mean that they are that closely related, at least compared to the grand scheme of things. You can, again, have a lot of changes in that family. That order could evolve into many, many different subfamilies with completely distinct behaviors and or genetic information. Obviously, those subfamilies are closer than, say, classes, the classes between different animals, but it gets more complicated than this is all I would like, just would like you to know. And so you've probably seen phylogenetic trees before, but I just want to go over how we analyze them and think about them. And so generally speaking, they're drawn as these branching groups. And so in this case, we're looking at very, very broad groups, right? We're looking at lungfishes, amphibians, mammals. Good question. Oh, yeah, you're totally right. I'm so sorry. Yeah, this is backwards. I'm so sorry. Someone just mentioned, yeah, it's genus first and then species. So genus is capitalized. I'm so sorry about that. I'm going to fix that right now. Nope, you're totally correct. It is genus and species, yes. Sorry about that. Am I sharing screen again or no?
No.
There we go. Right, so completely correct. Genus and then species, capital letter on genus, lowercase on species. And so when we're looking at these different groups, these trees are trying to tell us how they're related to one another. What we need to remember, though, is if you think about, again, the fossil record, right? Fossil record, we're missing a lot of intermediate organisms, right? You just don't, you know, as we talked about genetic change and evolution, this is not something that is, you don't just wake up, you know, or an egg doesn't just hatch as a completely new, brand new species for the most part. Obviously, you have those, you know, random genetic changes where it's extreme and something survives, such as, you know, chimps and humans. But generally speaking, this is a transitional thing that occurs. It happens slowly over generations. And so slowly you'll have these different changes that pop up in genomes. And so let's start here at the bottom right where it's simplest. So let's talk about ostriches and hawks and birds. And so when you look at a phylogenetic tree, sometimes these branches mean something in terms of their length. So sometimes a longer branch means that species has been around for a longer time. So it'll be proportional to, you know, every inch of that phylogenetic every inch of that phylogenetic branch means 1,000,000 years or something along those lines. In a lot of cases, they aren't, unless they say specifically. Often the lines just mean relations, and so their proportions don't necessarily mean anything. But this dot here that we're looking at 6 down at the bottom, what that represents is what's known as the common ancestor. It's an organism that most of the time we don't know exactly what it looks like or what it is. It's some type of transitional period between species. And so at some point, ostriches and hawks came from some ancestor that had some mixture of their two traits. And so when you're looking at these, you can say that crocodiles share a common ancestor with birds, right? And so all birds have some traits that are found in, or crocodiles have some traits that are found in birds, birds and crocodiles. And so back here is some hypothetical organism that we haven't found that is the common ancestor that has some traits of crocodiles, but then started to say, you know, evolve into birds, started changing their circulatory systems and feathers and things of that nature. Scientists also like to denote where specific traits arise because sometimes that's the easiest way to think about it. So for instance, we're not exactly sure where the most common ancestor is between lungfish and amphibians, right? When they started to walk out onto the earth, get limbs. And so what scientists will do is they will put these hash marks to denote the evolution of a brand new trait. And so for instance, this is pteropod limbs. So at some point, they had a common ancestor that didn't have any limbs. It continued to be a fish. And then at some point, it started changing and it started getting limbs. And so generally speaking, unless you are told otherwise, these traits that are shown with hash marks are held by all organisms down the line. There's something that exists in the genome and they have that. So if you look at, you know, all of these, you know, limbs, most organisms have those limbs, right? They have those pteropod limbs. So it's something that's held by everything after it. Obviously, there are changes, you know, organisms will lose things. So for instance, right, you know, pteropod limbs evolved millions and millions and billions of years ago over here. Snakes do not have those limbs, obviously, right? And so there can be changes in this history. And that's part of why this gets more complicated, because if you were looking at straight morphological traits, theoretically, snakes could be somewhere over here, right? They don't have limbs anymore. Perhaps they evolved before limbs and split off in their own way. And so scientists, when they look at phylogenetic trees, one thing that you need to know is, you know, phylogenetic trees are hypotheses. They are good guesses, especially nowadays. We're using mixtures of morphological traits and genetic traits, but they are ultimately hypotheses. If you ever read a paper that is fully looking at phylogenetics of a group, you're going to see that they're going to give you more than one phylogenetic trait. They're going to give you 10. They probably made 200 versions of them. And so what they're going to do is they're going to tell you the one with, given their data, what seems to fit the most of the evolutionary history in those changes in genomes. And so, you know, this evolutionary lineage is a sequence of organisms leading to a particular, you know, descendant taxon that we can look at. And again, a lot of this information is from fossils, right? These are organisms that we've seen in fossil records, and we're making estimations on their life history based on those physical traits that we can see and or genetic information. And so a branching lineage shows that these organisms are becoming different taxa. And so, for instance, you have these idea of sister taxa. So a sister taxa, when you're looking at a group, is the closest related organisms to one another. So for instance, looking at taxa B and C, or looking at taxa C, the sister taxa would be B, because they share a common ancestor at 4, and they're the closest related to one another. And so that would be, again, equivalent of chimps and humans, right? The closest thing that exists still in the wild is chimps. We are closely related to them. We have some common ancestor we share with them. That being said, this is why I said that you need to pay attention to context, because the closest, sister taxa of this, obviously being C is that, but the sister taxa of A, what is that? There it is. It is this entire group of organisms. So this is why you have to pay attention to context of groups. And so this is taxa A. This may be like great apes, or maybe some considered to be lower level monkeys or something like that. But what's going to happen is that the closest group to them will be the entire taxa that incorporates both humans and chimps. And so again, you need to pay attention to what they're talking about in order to know. Because again, when we look at, say, all of the, let's say, we'll go really far back. Let's say this is all mammals. What is the sister taxa of the mammals? Well, you have to look at this common ancestor and then see what group this D, E, and F is. So like the sister taxa of this whole group is this whole group down here. And so I wish they would use different words in order to like label these, but it would get very complicated themselves. You have to memorize a lot of vocabulary. And so when you have something, say like this poly break, you have some unresolved pattern of divergence. And so what that means is we're not sure if all of these species evolved at the exact same time, or if these species, it is possible that D came first and then it's split off into E and F, right? That's something more like this, but we don't know that for a fact. And so we're putting these lineages at the same level. But again, these are hypotheses. Until we find out more information about those taxa, we're not going to be sure about that. Another thing to note is if you look at, say, these chimps and humans, this little arrow is showing you, these can all be reversed. So the order that you put B and C in doesn't matter. This could be, it means the exact same thing, evolutionary speaking, if this is humans and this is chimps, or this is chimps and this is humans. These are complete, all of these branches are reversible and turnable. So for instance, you can also say, take this whole taxa and put it here, and then take this one and put it up here, right? They're all interchangeable in terms of their organization. What really matters is where these common ancestors fall, how they're related in terms of that. And so, so basal taxa, sorry. So the basal taxa, generally when you're doing a phylogenetic tree, you have to have what's called an outgroup. You need to have an organism that is very distantly related to the other. And so that's usually called the basal taxa, which is a comparative group that diverged a long time ago. And so you can kind of place your species relative to this one. So what you'll know is this basal taxa is very, very far related from these. And so they have a common ancestor, but you're mostly interested in this. And the reason we use basal taxa when we do a lot of things with computers and phylogenetic creation is if you are doing these hypotheses, right, you're putting this data into a computer to build a tree. If your basal taxa that is very distantly related, if you make a tree and that basal taxa, let's say G, comes up in here, and it seems like it's very closely related to B and C, there's something wrong with the way you're making your tree, because that this species should be very far distally related. And again, that's why these are hypotheses. And so there are a lot of variations in how these trees can be shown. There's this idea, what I've shown you this entire time is known as a vertical tree. So these branches slowly start going up. But this information is exactly the same as the information that's shown on the right here in this diagonal tree. No matter how it's drawn, again, the most important thing is you're looking at where are these common ancestors. So either way, chimps and humans share a common ancestor. Lizards share a common ancestor with chimps and humans. Or even more so, if you want to be more specific, lizards share a common ancestor with the common ancestor of chimps and humans. Again, this is that when it comes down to the debate, there's always a missing link. Since everything is a transitional state, we're always going to be missing some organism there. And so we need to look at it in that way. Yeah. And so an important note in this is that, taxa aren't a sequence of evolution. They are not like this creature became that creature, right? There's a common ancestor that is splitting off from them. And so we're not saying, that lizards became chimps. What we're saying is that some common ancestor started changing and became a lizard. And then that common ancestor also started changing and became what would be the common ancestor of chimps and humans, right? This is not, I guess to be a nerd about it, it's not like a Pokemon type of evolution. One species is not turning into another species. Those species are diverging like those, say, different birds, and they're gaining different traits. And eventually that leads to them being unable to mate with each other and leading to those differences. Yeah. And so this is just another example of if you change just the orientation of all of these by spinning them, you can make a tree that looks like this, but it's telling you again the exact same information. These trees are the same in terms of their relationships between everything. So again, I think it's easiest if you put a dot at each of these intersections and you look at it that way in terms of which species are related to others. Because in both cases, fish are the most distally related. Once again, if you were making this tree using morphological traits or genetic traits, and your fish all of a sudden was more closely related to lizards, you have a problem. And so this is an example of what I was talking about in terms of branch lengths mattering. So sometimes branch lengths can mean time that a species has existed. So for instance, again, Drosophila is the most the most distally related, has existed on the earth for the longest time in this one. So it has the longest branch, whereas, humans and mice evolved more recently. Generally speaking, it will continue to the end. I think it's weird that these end here because generally that means like that species is gone. So I'm pretty sure we're still around. Which is a little weird, but the length matters in that way. And then also specifically in this one, they're talking about how it could be genetic change. How much genome difference is there between each of these organisms? And so once again, flies, the Drosophila, fruit flies, have the largest genetic difference from this common ancestor and these other organisms. And so that's why in papers and stuff, figure legends are really important. Because looking at this, we have no idea whether or not if we didn't have that figure legend, we wouldn't know whether or not the lines mattered at all, if they were showing genetic change, if they were showing just time that a species has existed on Earth. And so they may or may not indicate that degree. So as I was saying, in the beginning, we did all of these comparisons of phylogenetics by looking at morphological traits. For instance, these are all the major spider families and just their faces. And we can see that when we compare spider families, they have very specific traits that are held by them. So for instance, these are jumping spiders. So all jumping spiders have the same breakdown of eyes. So in this case, if you don't like spiders and you don't like they have 8 legs, maybe you also don't like to know that they also have 8 eyes for the most part. And so spiders, you can tell how distantly related they are by their placement of their eyes. So if you look, all of these black dots are different eyes, different sizes and placement. And so jumping spiders can actually see almost 100% around them. I think they have like 15 degrees directly behind them that they can't see. But with the placement of their eyes, they have almost 360 vision, at least mildly. You know, the eyes on the back and stuff are mostly from motion. But that's why if you ever try to sneak up on a jumping spider, if you're brave enough, they will often turn to you and stare at you because they can see motion in their back eyes, and then they're turning to see you with their really good eyes here. And so this is how we used to do things solely. We used to look at morphological traits, and now we're looking at genetic traits. And so one thing we notice is if you look at, say here, this is the ogre face spider, it also has these giant eyes. So back in the day when we were looking at these different species, you know, these hunting predators had more similar features. And so we thought that these two were more related than they were based on the fact that they're both hunting in similar ways and that they have different eyes and things of that similar eye placement, you know. But after looking at genetic features, we actually realized that jumping spiders are a completely different group. They branched off millions of years. And so this is actually, the ogre face spider is actually an ancestral form. The ogre face spider uses silk and uses a web to catch stuff. The jumping spider has decided to lose its web completely. And so that's a change that we've seen only with the enhancement of genetic information being added to it. And so one thing we have to deal with, though, is this idea of homologies. And so these are physical and genetic similarities that are shared to an ancestor, right? And so all spiders share a trait that is they all produce silk. And so they have a homology of silk production that exists in all spider species. And so there are a lot of these throughout evolution. You know, one of the most classic ones that we talk about in terms of homologies are bones within the mammalian hand, right? If you look at mammals, we've all evolved to do very different things. Humans' hands to grasp are very different than cat claws for jumping or for whales and swimming. But if you break down the bone structure of all of these different hands, you'll see that they're made up of very similar breakdowns, right? They all start with a humerus, some type of connective muscle that goes into the body cavity. And then that humerus is connected to 2 different bones. If you look at this, in every case, the humerus is connected to 2 different bones. Obviously, their placement, size, and shape has been adapted to their environment. But they still have that same structure. Then you have carpals, these smaller ones, and then leading into metacarpals, and then eventually into your digits. And so all of these have a evolutionary base that we can compare, right? We know that at some point these came from some ancestor that had a similar bone structure before it started being specialized for each of these different environments, for swimming, for jumping, for flight. And so homologies exist on many different levels. Again, use of silk, production of silk in spiders is another homology that we can track along evolutionary lineages. All right, so it is 10:15, so I'm going to let you all go. I'll continue with this. Next class, we'll start talking about how these homologies have to do with actual changes in our DNA and how we can look at those changes in order to see these differences. I'll go over how we align sequences and how those blast searches that we did in class to look at species matter.
Thank you.
You're welcome.
Thank you.